Hey guys, so you were having some trouble with the models pertaining to embryonic and fetal development. So here I'm going to go through it with you using these models and of course also microscope slides so that you guys can see the overall picture with regards to development of the embryo and fetus. So let's go ahead and let's get started. So of course we're going to start with fertilization, which is also known as conception. This is the fusion of the male and female gametes. It's actually a really in-depth process, and I go over the process in the male reproductive video, and that's at a bare minimum. I don't really discuss it at length, but it's a really in-depth process. But here we'll just go through just a quick overview of it. So what happens is the acrosome of the sperm cell that I have arrow pointing to at the top there, it fuses with the corona radiata, and then it releases these enzymes from the acrosome that interact with the zona pellucida of the ovum. Okay? And then what happens is that these cortical granules within the zona pellucida fuse and they harden the plasma membrane so that prevents polyspermy. So only one sperm can fertilize one egg, and of course once the sperm makes it through the zona pellucida, you have fertilization. And you can see in the two models here, I have labeled the acrosome of the sperm head, and I also have an arrow pointing to the sperm attempting to fertilize the egg in the fallopian tube, because fertilization takes place in the fallopian tube. So once fertilization takes place, the ovum or the egg becomes the zygote. And you guys can see I have a microscopic image of a zygote here, and then of course an arrow pointing to the zygote on the model. So the zygote is just a fertilized egg. So what happens to that zygote is that it undergoes cellular division. So you'll start to see cleavage of the zygote into a 2-cell stage, 4-cell stage, and 8-cell stage. And the cells that are formed from this cleavage of the zygote are called blastomeres. So the cells are called blastomeres. And I just want to point out that the cellular division is actually occurring within the fallopian tube. Implantation has not occurred yet. So once the zygote has undergone numerous cellular division, it becomes this solid ball of cells or blastomeres, and this is called the morula, still within the fallopian tube. So eventually the morula develops into what's called the blastocyst, and the blastocyst is just this hollow ball of cells, and if you remember, the cells themselves are called the blastomeres. I do want to point out that it's once the embryo reaches the blastocyst stage, that is when implantation takes place. Now in mammals, the blastocyst is actually referred to as the blastula, and so that's probably what you'll hear me refer to it as for the rest of this video because I learned it that way teaching freshman biology. So parts of a blastula include, for instance, the fluid-filled cavity called a blastocele that surrounds the blastomeres. There's also what's called the embryoblast, or the inner cell mass that gives rise to the fetus. And you can also see the trophoblastic cells, that give rise to the extraembryonic tissues, such as your chorion, your amnion, and your allantois. So eventually the blastula is going to enter the gastrula stage, and this is of course when gastrulation is taking place. So now we have this hollow cup-shaped structure, and the model below isn't correct because we should be seeing this invagination of the cells to form the germ cell layers. And so on the microscope slide, you can see that invagination of the blastula. So once you see this invagination, it has now become the gastrula. Okay. You also see this invagination forms the blastopore. In humans, that blastopore becomes the anus. Okay. You also see the archenteron, which is the other end of the invagination. And then it's here in the gastrula stage, once again, where you get that differentiation of the germ cell layers. And your germ cell layers are your ectoderm, endoderm, and mesoderm. You don't see the mesoderm here, you just see the endoderm and the ectoderm. This is an early stage gastrula. I just want to point out that humans are triploblastic organisms, so we have the 3 germ cell layers, but other organisms such as your jellyfish, which are your cnidaria, they only have 2 germ cell layers. So here, once again, you see the ectoderm and endoderm, and this is an echinoderm, so it is a triploblast. The mesoderm just hasn't formed yet. So this is just going into the fates of the different germ cell layers. Your endoderm eventually becomes the lining of your digestive tract. It's also found in your liver. Mesoderm you can find in your lungs. It gets differentiated into your skeletal muscle, cardiac muscle, red blood cells. Ectoderm forms most of your outer body structures, such as hair and your skin cells, as well as your nervous system and your face. So those are the fates of your germ cells. Now, what's the fate of the embryo? So, right, those trophoblastic cells, they differentiate into your extra embryonic layers, so your amnion, chorion, and allantois. And so we're going to go ahead and we're going to look at different parts of what's called an amniotic egg. So we are amniotes. We make an amniotic egg. It's just that unlike turtles or lizards, we don't have an actual leather outer shell for our egg. So starting with the amnion, it's actually in this model. It's just, once again, not the best model. But I have it pointed to with all these arrows at different stages of development. So this amnion is what encloses the embryo, and it fills up with fluid and becomes the amniotic sac. And in essence, what the amnion is doing is that it's protecting the developing fetus, it's preventing desiccation, for instance. There's also the allantois. The allantois is just an outgrowth of the embryonic gut. It basically connects the fetal bladder to the yolk sac, so it's where waste is removed, right? Because the fetus obviously can't remove its own waste. And this is why the allantois forms part of the placenta, right? The allantois and the chorion form the fetal portion of the placenta. And the reason why they do this is because the fetus, right, cannot breathe, and its heart is not necessarily functioning in that way, right? It's not sending blood to the lungs yet. And so in order for the fetus to get oxygenated blood, the fetal portion of the placenta has to combine with the mother's blood vessels so that the mother oxygenates the blood for the fetus. The allantois has to converge and form portions of the placenta as well because the mother has to remove the fetal waste, right? And of course, the chorion is the outermost membrane surrounding the embryo. Once again, it merges with the allantois, forming the chorion allantoic stalk. This is the fetal portion of the placenta. And those chorionic villi that span out from the chorion allantoic stalk are very important because they provide maximal contact with maternal blood vessels. And once again, right, the mother has to oxygenate the blood for the fetus. And last but not least is, of course, the yolk sac. I have it pointed out in all the portions of the model here, the developing yolk sac. And this is just to provide nutrients to the developing embryo, right? And of course, the chorion allantoic stalk is also providing the embryo with nutrients from the mother as well. And so that is it for embryonic development, embryonic and fetal development.